from Microbe TV. This is office hours for Wednesday, June 18, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet, where we can talk about viruses or other things as you wish. Welcome, everyone. Good to see you all again. Who's here? Let's see who's here moderating for us. We have Steph SF. We have Vanity Nutrition. We have Les. We have Tom Steinberg. Thank you for moderating tonight. On the agenda for tonight, we have some news, as always, and a mini lecture. Emerging Viruses Part 1. You're going to like that. Everybody's into emerging viruses, right? Let's see who's here tonight. Tell us where you're from. We'll shout you out for 20 minutes or so before we get started. Wayne is in Urbana, Illinois, where it is 75F, 24C, clearing up after a stormy day. Welcome. And Steph is in San Francisco, 18C, sunny and windy. Will is in Changsha, 25C and cloudy. And Changsha has its lowest U.S. air quality index in 11 years. Very good. Congratulations. And congratulations are in order for Vanity. I don't see her. Here you go. Big news on Monday. Was accepted to medical school. There you go. Now your feet will truly be on the ground. And I hope, Vanity, that you vaccinate everyone and you do not give medical exemptions when they're not called for. Promote vaccination. I'll be curious to see how they treat vaccination in your medical school. Let us know, okay? Costella is here. Hello. Like Iowa, Ohio, something like that. Mark Martin is here. Tacoma, Washington. Visto is in 10C, Sydney. Yeah, it's winter down there, man. Hey, we had winter. We just passed through it. Now it's your turn. Mark is in LA for ASM Microbe, 28.5. I used to go to those all the time. Not going anymore. Coralie is in New South Wales, Australia. Pete is in a sticky inner London, with a pair of urban foxes making interesting loud noises, like mating noises type of noises. Tom is in Oregon, in the Coast Range. Welcome. Mike and Lori Dinger in Seattle, 20C and breezy. Happy birthday, Mark Martin. Not a big deal anymore. I know that. Yep, I don't care about my birthday anymore. Very Nika, Privyet. Wow, you're in time. Dobry večer from Moscow, where it's cold again. I always miss lives when it's actually hot, so every time I'm here, I'm proving stereotypes about Russia. 13C and rainy. Welcome. Nice to have you. Kang is in Chicago, where it's 19C with intermittent rain. Welcome, Sandra. Was from Mexico, but now somewhere else. John is in Minneapolis, 26C. Nevitz, 3 more days to summer solstice and maximum daylight in the northern hemisphere. That's right, the 21st. The longest days, and then it's all downhill from there. Before you know it, the summer will be over. I enjoy summer very much, but it just, boom, it passes in a flash. Oh, well. Julie S. is in central Pennsylvania, 84 Fahrenheit. Storms on the way. What is it here? Let's see. It's raining here when I got out of the car. Weather. 25C and cloudy. Humidity is pretty high here. It is 86%. Darn high. John? Well, we already saw John. Yeah. Krishnan, good morning from Malaysia. Good to have you back. Logotic is in Chicago, where it's 22C. Steve is from Paso Robles. 99 degrees. Oh my gosh. Paso Robles, of course, is the home of a eponymous vineyard, which has very nice wines. Jonathan is in Encinitas, 23.5C, warm ride from the lab. He's always driving home when we start the office hours. Our favorite nephrologist, Carol, from Bakersfield, where it is 36C, quite hot there as well. Just so everyone knows, a nephrologist takes care of kidneys, okay? You will definitely advocate for vaccines. It's good. Rona is on Long Island. Just returned from Vancouver. Very nice place. Laura is in Washington, PA, 26C, hot and humid. Javier is in southern Ontario with a cloudy and humid 22C. And with my final exam of high school over with. So now I know you're a high school student. Well, you were. I didn't know that. I could not guess from your speaking. So good for you, speaking like an adult. I guess you are, right? You're over 18. But you know what I mean. Foxes, unusual mating or fighting, who knows? They should have growing cubs to look after now. Silent runners in Wichita, Kansas. 2.4 inches of rain, 90-mile-per-hour winds yesterday. Not unusual for Kansas. Abdul Rahman from Manhattan. Abdul, you were supposed to visit the incubator a couple of weeks ago. You didn't come. It's okay. Something happened. Leo is on the Mendocino coast, 16C. Polka Dot Pup, North Texas. Cold morning, hot in the afternoon. Alaska. Well, Terry is obviously from Alaska. We have 24 hours of daylight right now. Wow. That's right. Tomorrow is Juneteenth, a holiday that the president is trying to get rid of. Just a disgusting show of behavior, right? Folks, I don't know how you can vote for someone like that. I don't think anyone here did. Peter S. is in Boulder, where it's 26C. Rainy is in near Raleigh. That's good. 90F and muggy. 3 a.m. in Moscow and already light. EHS Talent is in Lehigh Valley. Calm post thunderstorm. My son is in LA at the conference, was hoping to meet you. Sorry. Now, I have a variety of reasons which I will not elaborate on for not going to ASM Microbe anymore. Mateo. Where you from, Mateo? You have to tell me where you're from. And my favorite virus is poliovirus, which I spent most of my career working on. So it's an amazing virus. We did a lot of cool stuff with it, and it has fascinating properties and continues to fascinate through the efforts to eliminate it. Notice I say eliminate because you cannot eradicate it. What's my favorite wine? I don't want to go to Miami Beach this winter. Prump. I'm here all week. Try the veal. There's a joke about that. My favorite wine, I don't know. I don't have, I have some favorite French wines that nobody can afford, right? I really love Pomerols, those reds. But if you want to, I like the Paso Robles reds. They're really fine. What's it? Dow, D-A-O-U, I think. Let me look it up. Yeah, it's a Cabernet, D-A-O-U, made by Paso Robles, and it comes in different price points. The more you pay, the better it is, Mark. You need to drink less spirits. Remember those drinks we had made at the last ASM micro when you were having some get-together at a bar? We had some nice drinks there. I asked them to make something with rum, which was unusual, and they made it for me. I enjoy Michael Osterholm Update, who always relates the daylight to a pub in Auckland for comparison. Makes me smile. That's very nice. That's a good way to do it. I didn't know he had a sense of humor, because to me he was always nasty. Can you comment on this goofy idea that the latest COVID variant presents with razor blade throat? You know, a handful of people get a particularly bad sore throat, and now it's a thing with the variant. You cannot conclude that the variant is causing this. You have no idea what other factors are playing into it, right? It's just crazy. But this is COVID, right? Everything is all verkakte. Kip and Laura, thank you for your contribution to science communications. We're here to learn more about bugs and drugs. Yeah, Chateauneuf du Pape is okay. I like full-bodied red. Oh, I forgot the Italians, the big Tuscans. Let's see. Yeah, that's going to be my favorite. Big Tuscan red. Let's see a name. Let's get a name. We're going to break your bank. You know, the big Tuscans include like Cabernet and Sangiovese, Brunello de Montalcino, Chianti Classico, Sassicaia, Tignanello. Those are all great super Tuscans, okay? Can't go wrong. But these are full-bodied wines. I prefer that. Chateauneuf du Pape is fine. I didn't get an acceptance? What do you mean? I told you could come. Make another date next week sometime, okay? Because I'm away Thursday and Friday. No problem. Areozone from Asheville, North Carolina, 25C. Vanity misses flying to Moscow. Well, you're not going to fly anywhere anymore. That's it. You're done. It's very nice that you're going to record episode 99. But yes, you share many of my concerns. It's okay, Mark. I don't mind. I don't expect everyone to follow what I do. Okay. Leo has to plug his local product, a nice Pinot from Anderson Valley in Mendocino. Mateo is in Bogota, Colombia but now lives in Glasgow. Terry likes Alaska blueberry wine or fireweed wine. Patricia, hello from Massachusetts. Michael, Mexico City with rainy days and probably more so since Hurricane Eric is close by in the Pacific, 16C. And I wanted a rum old-fashioned because I think I started with an old-fashioned with, with whiskey in it. And then I said, could they make a rum old-fashioned? And the bartender climbed up to get a special bottle of rum. Yeah. Mary is in Corvallis, 18C, and clear. My first time live. I don't know if I can rise to the level of banter. Sure you can. Just by saying that, you just rose to the level of banter. Glasgow is Scotland is very Nika's dream. That's where Daniel Griffin was last week. Yes, Christina Nowla from TWIP. This is great, Kang. I should have sent the photo from No King's demonstration where there's a dressed skeleton holding a sign, I believe, than RFK Jr. That's precious, Kang. I love that. It's really good. Why did it make Super Tuscan? That's what they're called, right? Super Tuscans. They're really full-bodied reds. Ms. Martian is from Mexico. Welcome. Multiple people from Mexico here. Pete had a Montepulciano as a house red, celebrating a friend's 60th this week who doesn't drink. For what it's worth, the scientific term verkakta is Yiddish that generally means messed up, ridiculous, or defective. Yep. I had lots of teachers of Yiddish over the years, from college to graduate school to my faculty appointment at Columbia. People always taught me. And my chairman, Sol Silverstein, had a copy of that book by Leo Tolstoy, I think it is. What is it? What is it, Leo? The Joys of Yiddish by Leo Rosten. An explanation of the Yiddish language and its influence on English. Yeah, the Joys of Yiddish by Leo Rosten. He had that in his office, and he'd also pull it out and teach me a new word. Thank you for donating, SRO. Here it is. Thank you for your support of science communication. Really appreciate it. We need everybody's help so we could put the record straight and put real science out there, because what the administration is doing is lying left and right. The latest lie, well, we talked about this last week. RFK Jr. fires all of the ACIP, right, the CDC vaccine recommendation panel. He says they're all have conflicts of interest. They don't. It's a lie. And he has the biggest friggin' conflict of interest. He takes money from a law firm that he works for to help them litigate against Gardasil, for example. How can you be Secretary of HHS and do that? No morals whatsoever in this administration. Zero, from top to bottom. Polka dot pup. Do you think the fall vaccine would be appropriate? I don't know. There's been enough hints from Macari and Prasad that it'll be okay. The thing is that they're not going to recommend it for under 65 or pregnant women, which is the dumbest thing in the world. Both recommendations, okay? First of all, anyone who wants it should get it. It's licensed right now. But they're going to de-license it for pregnant women, which is a risk factor. Pregnancy is a risk factor for severe COVID. Every other country in the world knows that. This is just malignant ignorance. People are going to die. You wait. The first pregnant woman who dies of COVID, I'm so sorry to say that, there's going to be a lawsuit against RFK Jr. I don't want it to happen, but there will be. Anyway, I think COVID will be okay. Now, the problem is, folks, next week, I think next or the following week, ACIP is meeting, and the agenda's out there. They're going to talk about MMR vaccine. I'll bet they de-recommend it. The bastards. And how many kids are going to die of measles as a consequence? They have no morals whatsoever. They're insane. Should we all be chanting Vincent, Vincent like a big rock show? Why? I mean, so you can. It's fine. I do like doing live events. And 2 years actually, we made a T-shirt, a TWIV T-shirt, and on the back was a list of all the shows we've done. We filled up the whole back because we went to so many places. I loved doing that. That was so much fun to meet people who are fans. We have to be careful about using bacteria for forced labor. So what Carol is referring to is a paper we did on TWIM last episode, where the authors of the paper, this is a paper on the Gowanus Canal. They dug up sludge and they're looking at the microbes out of there. And they found some that could be used for bioremediation. If you have some contamination, you could put those bacteria in. But they said we have to be careful about using bacteria for forced labor. I couldn't tell if it was a joke. Come on. We use bacteria all the time in the laboratory. We make them do work for us. They're not sentient. Okay, I could see a mouse or a rat or a guinea pig or a dog. Fine. But bacteria? Next, we're going to talk about water molecules, making them work for us. Thank you, Carol. Terry is at China Hot Springs. He promised to talk on bioterrorism fungus. It's in TWIM, which is going to be published Friday. Okay? It's in TWIM. That's what I meant by that. Not here. Today's theme is wine. Yeah. So you just have to start one thing early and it catches on. Yeah. Mark is in Vegas. Over 38 until Halloween. Yeah. Jeff is in Elgin, Illinois, 70F and raining. El Tuavino Veritas. There you go. Vista, I heard some real experts are creating a private vaccine advisory board to rival the RFK Jr. conclave of clowns. That's good. Will this work? Yes, I have heard this as well. So Michael Osterholm has already started one, and there are other people who are starting them. I don't know. It depends. I mean, if they put good people on, which I think they will, then they can make recommendations and the states can pick up on them, because the CDC is going to be crazy. Nev, it's a good No King's March sign I saw in New York. Don't make me repeat myself. History. Yeah, you know, we had this thing called a Revolutionary War in the U.S. to get away from the Brits, you know, taxation without representation. We want to be ruled by a king. Yeah, what's going to happen? Now, we can't have a Revolutionary War because this guy will just whip out his military and crush everything and then declare martial law, and that'll be it. One of the last Russian phrases I'll ever forget is this. I rode the metro. What is it? Something like, stand clear of the closing doors. James from Brisbane, Australia, welcome. For a great Yiddish sub chat, we need to get Amy for sure. Absolutely. It's about 18C in Brisbane. Abdur, who benefits from cutting Gardasil? Nobody benefits from cutting Gardasil, but RFK is involved in anti-Gardasil litigation. So if he takes it off, then the case for the litigation gets stronger. So the lawyers benefit. There you go. There's no severe side effect of Gardasil. They're all lying. Every one of them is a liar. They don't know any science. It's disgusting. And I can tell you that because I spent many years in science. And some of these anti-vaxxers have spent many years doing what they're doing. But sorry, you're wrong. Hello, Joel. Thank you for your contribution to science communication. Really appreciate it. Thank you for your courses and content. My pleasure. I love to do it. Pete lost track of one of my stupid carry-ons. Did the U.S. ever share its flu surveillance data to WHO for this year's vaccine flu strain selection? I don't know. I really don't know that. I doubt it. What do you mean you lost track of one of your carry-ons? What does that have to do? Did you ever see this Instagram video where they have videos of people in different airports handling luggage? I mean, in every country, they just throw it all over the place. But in Japan, they carefully put it on the conveyor belt very nicely. They don't throw it. And in China, they even wipe down the bags as they're going around the carousel. Why can't we do that here? Mark, HPV-driven cancers will benefit from Gardasil. That's right. So people will die of cervical cancers, anogenital cancers, oral cancers, because they're not protected using Gardasil. This is a crime, folks. Mary Nika says, hey, speaking of viruses, a British lady died of rabies because a stray puppy from Morocco scratched her. I remember the same case in Russia. Another reminder to never touch wild animals, no matter how cute. Yes, that goes for all of them. It was 100% a joke about bacteria. What? What Vanity said? I don't know. You need to give us more data here. Lots of bacterial forced labor in the vinegar industry. Yeah. Cruel but tasty. Oh, all kinds of industries. Vinegar, wine, cheese, any food product that's fermented, right? Howdy, Rita from Idaho. Yeah, we use yeast to make alcohol. I saw some bacteria protesting with no Listerine signs. Forcing bacteria will be considered a gain of function. Oh, speaking of that, the administration just said it's going to crack down on dangerous gain of function experiments that are currently in process. They're going to stop them. Idiots. Pure idiots. Polka Pup says, this sounds nuts, but the government says they are not cutting Medicare or Medicaid. Right, they just withdrew health care. They'd like to if that big, beautiful bill goes through, right? RFK can start a movement for cancer cell rights and virus rights, too. Stop interfering with natural evolution. Well, he doesn't believe in infectious diseases, right? Ah, the doors are closing. I thought so. Yeah, I like hearing that in different languages in different countries of the world. The doors are closing. Yeah, I love that. No one talks about the fungal labor in cheese industry. Kevin is in San Francisco, 71F. Joel is in Bath, UK, where it's sunny and warm. Big cancer benefits from people not using Gardasil. Elizabeth says the military are not widely behind the orangutan. At best, he has ICE and a few militias, maybe some of the FBI, but not all. A war at home would not go his way. I don't want people to die, right? But he's the commander-in-chief. Doesn't he have the military at his behest, right? I don't know. Yeah, it's either ignored or a very sporadic discussion for people not talking about forced labor of fungi in cheese. How do I think the thimerosal discussion will go next week at ACIP? Well, the only vaccines that have thimerosal are the multi-dose flu vaccines. It's out of everything else. We want to take it out of that? Fine. But you can't have multi-dose then because it'll get contaminated with bacteria. And then you'll give people sepsis. Okay? So you have to do single-dose vials. And so as the docs know, it's really, if you're doing a lot of immunizations, it's nice to have a multi-dose vial. So I think they're going to can it. It's a religious group or cancer drug company against Gardasil. I don't know what you mean, but they're, these law firms tell people, if you got damaged by Gardasil, come see us. And then they sue. I don't know how you got damaged by Gardasil. Because, for example, if you got Gardasil and then the next day you fell and broke your leg, that has nothing to do with Gardasil, but they will say, I broke my leg after I got Gardasil. Polka Dot Pup says, if the government withdraws medical care from the elderly and disabled, does Medicare and Medicaid benefit? They don't have to cut the programs. Well, that's what they're cutting, Medicare and Medicaid, right? Because they think there's a lot of fraud going on. I don't know. I just don't know. Yep, in London, it's Mind the Gap. In Spanish, it's like manténganse alejandro de la puerta, something like that. Correct me, folks. I used to hear that all the time. The duty to disobey. There you go. Not really on the military side. If orders are unconstitutional, they can be refused. Plus, it's an unwritten code that the Joint Chiefs is very much against internal military action. Yes, that's correct. You're not supposed to use the military for domestic stuff, right? Duncan says, hi from North Wales. A couple of weeks ago, I saw controversial cardiologist Asim Malhotra on the C4 TV channel proudly recruited to RFK Jr.'s department as an independent healthcare advisor. All these, supposedly credentialed people that don't know anything. Yes, Koldorf testified against Gardasil at trial. That's absolutely correct. Yes, orangutan is great. It really is good. Elizabeth says, I didn't say it would suck. It would be illegal to order the military to attack U.S. citizens except under very specific circumstances. You can't just say it's an emergency. So what did he do with the National Guard? He sent them to L.A. where there was no need, and then they push people around, and say there's a riot. Twiv amazed me, John says, with the concept that our breath of our antibodies expands for a while, and it never occurred to me that it is necessary for that process to stop too. It's right. You can't be expanding forever because then you start to cover your own epitopes, right? Self-antibodies. Okay, we have a little pause here. So let me mark that. Let me do a couple of news items. Okay. Copy. I have to copy this one. It's the New York Times, and I'm not going to be able to see it on that computer. So let's turn on this computer. It's not going to be on this one either because I don't have my password on here. Here. I'm sorry. I shouldn't have done a New York Times thing. All right. This is a great article in the New York Times. A CDC vaccine expert basically is interviewed for this article, and she left the CDC because she said, I can't trust the CDC anymore. She used to give all kinds of expertise advice. She was at the CDC about vaccines. It was really great. And she quit because she said, I can't trust the CDC anymore. These bozos, I don't trust that they're going to do the right thing for people, and I can't be here while that is happening. And unfortunately, I can't get into this article here, but that's the gist of the article. It's very powerful. She's obviously smart and very well spoken. If you can get it, read it. Now this one I can get. This is an article in the New and Observer. Get out of here, all these ads. It's disgusting. I can share this with you. North Carolina lawmakers launch fact-finding mission into UNC coronavirus researcher Ralph Barrick. Okay. So, Ralph Barrick works on coronaviruses, and he has genetically modified coronaviruses over the years to see what's needed to infect human cells. And the House wants to get his lab records. Sued UNC for records as part of its campaign to collect more information on the origin of the pandemic. There's no origin of any pandemic in North Carolina, nor in what Ralph Barrick has done over the years. These people know nothing about how virology works. Donald Trump rejects scientists' claims that the strongest evidence says the virus occurred naturally. Who the hell is Donald Trump to reject this evidence? He probably doesn't even understand a word of it, and he rejects it. The White House website, which you've seen, contends that the coronavirus, SARS-CoV-2, was most likely born of gain of function. Barrick collaborated with the Wuhan Institute of Virology, with Zhongli Shi, and so they're saying that he enabled her to do it. So they're going to get the lab records and see by June 18th, today, we'll see what happens. I mean, yeah, they'll give them the records. Why not? There's nothing incriminating there. They're not going to know what it means. They're not going to know anything. They're dumbasses. Oh my gosh. So those are the two articles I had. Let's see here. In the meanwhile, some questions have poured in. The biggest health podcaster did an RFK video asking peeps to write Congress to toss RFK 2 million views. Let's hope. Okay. The guard isn't attacking. They're sort of acting as guards, but they're skirting the line. ICE is causing the riots. Okay, thank you. I don't want to talk about politics here because I don't know anything about it. And there's a lot of discussion. Yeah. The author of, thank you, Kevin, the author of the Times article was to present data at this upcoming ACIP, but refused to make the slides RFK wanted, and she quit. I don't blame her. Screw you, RFK. These slides are all lies and misinformation. How upsetting for Barrick, for the politicians that complain about witch hunts. They sure like to launch them themselves. Indeed. If RFK Jr. can convince people that fewer people died of COVID than the vaccine, he can convince them of anything. Well, he didn't convince everyone, of course, but a lot of dumb people, yeah, or people who can't understand. Tom says, with respect to North Carolina and lab records, they're not going to know what it means. Just the words crossing my mind. These lawmakers don't know anything about virology. Nothing. And they'll find that he had an email with Christian Anderson, and they'll say, that incriminates him, because they can't look at the science. They have no idea. Well, that's it, Mark. If they don't know what they're doing, they just make something up. That's exactly what they will do. They will make stuff up. Yeah. And they will they will take it from there. And Will says, since Zhongli Shi warned about the possibility of a coronavirus becoming a pandemic, she's not then going to create a pandemic. Well, not intentionally. What they would say, she had some bat samples that contained SARS-CoV-2. And she has said she did not have any such samples. She released the genome sequences of all the samples she had. And then people say she's a liar. But that's not science, folks. How about some evidence, okay? This is what this administration does. They lie, and then they call you a liar. Buggers. Yep, the hypocrisy is staggering. Really staggering. What Joel wants to know what I'm going to teach today. I'm going to talk about emerging viruses, concept about emerging viruses. It's going to be a two-part series we're going to do here and next Wednesday. And we'll start in a few minutes. So this video where they called for RFK's resignation was Dr. Mike, who John Elmaker likes. They make this stuff up. Cool, real facts and science, fake news, and the pathetic mainstream media will roll over dead. And of course, the real cause of a lot of the problems is Fox News, which just doesn't cover stuff like doesn't cover measles outbreak at all and misleads people, right? And so this is not what a news organization is supposed to be. I don't see why it can't be regulated. The regime, Ariozone calls it, is a hotbed of infantile and petty people. It sure is. Well, it's great that other people are doing this. For a while, I felt that we were, and I'm glad that other people, but people have joined, I agree, but they joined late. Okay, we were there at the very beginning. So we're talking about, just to clarify this, who said this? The biggest Raise 11, the biggest health podcaster, did an RFK vid asking people to write to Congress to toss him. So I don't know who did that, if it's Dr. Mike or someone else. Maybe someone could clear it up. So Abdur says it's the closest to reality, much more than any other medical show. Dr. Mike. But is it not clear that it's not clear to me that Dr. Mike did the thing calling for RFK's resignation? So maybe someone could clarify that. The vengeance presidency, that's for sure. This is true, Kevin. The new AI in use at FDA does not deliver accurate data so far, but it's being used to generate guidance documents as well as justifications for scientific decisions affecting 340 million Americans. You know, why do they have to use AI? They don't have brains. They can't do it themselves. Oh, it's too much work. I can't do it in time. It's tough. Then get out and get someone in who can do it. No, it's not ready for AI. Hello, Bad Cat. Welcome. Winthrop, Maine. You were at the TWEVO this afternoon. Good to see you again. Aha. Will says Fox News is licensed as an entertainment channel, so cannot be sued for fake news. Well, they shouldn't be allowed to call themselves Fox News then, right? Something should be regulating that. It should be Fox Entertainment. Then maybe people would get it. Nah, probably not, right? I'm giving people too much credit. But this AI thing really bums me out. Yeah, the Maha report of RFK Jr. was full of errors generated by AI, and the FDA now. Yeah, I've heard that there were lots of errors in documents. Hello, Charlotte. AMA gave an update today. I didn't see it. What did they say, Charlotte? Tell us. Or if anyone else has seen it, tell us. Oh, Vanity calls it Fox News Entertainment. Why do you have to have news in there? Because that's the name of the company? Fox News Entertainment. This is disgusting. I think this is a big factor in where we are today, Fox News. Oh, gosh. Let's do some science, okay? This is too frustrating. We'll come back to your questions. Faux news. Hello, Noir. Yes. It should be faux news instead of Fox News. Yeah, that's good. I like that. Yeah. All right. I'm going to... Elizabeth says they use AI because no one with any actual knowledge or ability will write the crap they want. That's right. Okay, let's leave the crap behind and do some science. I want to talk about emerging viruses. All right, let me see if I can move this. No, I can't move this bloody thing here. Oh, yeah, I can move it. There we go. Can I expand it? Yeah, I can. All right, emerging viruses. Nothing endures but change. It's true. Emerging virus. What is an emerging virus? Let's go back to the beginning and do some definitions of causative agent of a new or previously unrecognized infection. All right, so that's important. It's not just new viruses, but previously unrecognized, or as you'll see, a virus that now causes a different kind of disease. That can be an emerging infection. This is a term that arose in the 90s, but of course these emerging viruses have been around forever. Since the rise of agriculture, new infectious agents have invaded human populations because the populations grew as a consequence of agriculture. You know, before that, we were hunter-gatherers living in small groups of a few hundred people at the most because we had to catch our food, right? We couldn't grow it. So you couldn't have a large city because the city needs people need in the city to buy food because there's nothing to catch. So 11,000 years ago, you know, we figured out how to grow things and harvest animals. And so then people started living in greater and greater numbers. And that's when we saw viruses invading humanity from animals, right? And that didn't happen before agriculture and commerce. Now, recently, of course, we've gotten really good at detecting emerging viruses. We are really good at looking and finding things that have some potential to emerge, although now, of course, the administration has canned this one project that was tasked with, looking for pandemic threats, collecting them, by saying it was too dangerous. The inanity of that statement is absurd. You have to do dangerous things to advance. And as I said, an emerging virus can be a virus with an expanded host range and an increase in disease that you didn't see before. So an example would be Zika virus. You know, for most of its history, it was in Africa, and suddenly it got into Brazil and caused microcephaly, and we had not seen that before. So that was a new disease. All right, so the term zoonosis means transmission of a virus from a wild or domesticated animal to humans. A zoonosis or zoonotic infection or a spillover. Now, sometimes these zoonotic infections establish a new virus in the human population. So SIV moved from chimps to humans. SARS-CoV-2 moved from bats to humans, and they became human viruses. So that each infection is from human to human. It's no longer a zoonotic infection because it's just going human to human. It's not going animal to human. But often, human transmission cannot be sustained. So you have a zoonotic event which introduces Ebola virus, maybe from bats to humans, or MERS-CoV from camels to humans. And the chains of transmission are very short. And it remains a zoonotic infection because each human infection is a spillover. I love the example of West Nile virus, where every human infection is a spillover from a mosquito. So every West Nile case is a zoonotic infection. There's no human-to-human spread. So here's fever, which is a book describing the first zoonotic infection, Lassa virus, well, the first that we knew about and learned about in the 1960s. And this is a great book. You should read it. You can get it on Amazon now. I read this so I graduated from Cornell, and I had no idea what to do with my life. So I started working as a lab technician, and I found this book, and I read it, and I said, that's it. I want to be a virologist. So thank you, John Fuller, for doing that. It's a really good book. You can read it in a few hours. All right, so where do the viruses come from that we have today? Here is our best estimate. So it's a bar graph showing the viruses belonging to a number of genera. So here we have 31 genera. Viruses belonging to 31 genera. So like poliovirus is a genus. I'm sorry, enterovirus would be a genus. 31 viruses from 31 genera are human viruses. They transmit from human to human. They're not zoonotic, right? So they are probably originated in a zoonotic spillover. But now they transmit from human to human. So they're human viruses like herpes viruses and measles viruses and polio viruses. And yeah, SARS-CoV-2, it's a human virus now. 37 genera of viruses are zoonotic pathogens that spill over into humans and cause disease but do not transmit. Ebola virus, MERS coronavirus, Nipah, hantaviruses, and many more, some of which you'll see in this and next week's version of emerging infections. 16 viruses that are members of 16 different genera came from our Homo ancestors. So, we have Homo sapiens, Homo neanderthalensis, Homo denisovan, and maybe more. And our ancestors, like Australopithecus, which is shown here, they passed on viruses to us. So 16 genera of viruses came from Homo ancestors. And finally, 6 genera of viruses that currently infect us. That's what we're talking about here. Came from our Homo relatives. So we're Homo sapiens, right? It's Homo neanderthalensis, Homo denisovensis. They had viruses that they passed to us because we coexisted with them, for a while. Anyway, we probably gave them viruses too, but they're gone, so we don't know. So these are the origins of human pathogens. They all came from someone else. In no case has ever a pathogen, a virus pathogen, come from a laboratory. In no case. No case has a pathogen here on this chart. Nothing. There's no lab bar graph here because it never happened. And the people who think it happened now are fooling themselves. Just fooling themselves. I can't believe it. There's so much data saying the contrary. Anyway, what causes the emergence of infectious diseases? Let's talk about this for a little bit. The biggest driver is human population growth. Okay? We invade all ecosystems. We occupy every corner of the planet. It's our fault, folks. We did it. We caused these infections to spill over because we're invading everywhere. We invade areas where the viruses are, so we get them. And look at this population growth. Oh my gosh. In the last hundreds of years, it's just shot up. Look at this. It's incredible. And this is responsible for all the problems we have these days. I mean, it's just not just invading animal spaces and different ecosystems, but also human crowding. So we pass on easily viruses to one another. And we do lots of things to promote virus emergence, right? Air travel. Look at this. This is a snapshot from this website where you can see every airplane in the air at any moment. You can actually click on it and see what the flight is and where it's coming from and where it's going. It's very cool. But look at that. This is a main way that infected humans can travel, right? But other things, holding back water, irrigation, deforestation, rerouting of wildlife migration patterns, wildlife parks, long-distance transport of livestock and birds, particularly pigs, but birds as well, blood transfusions, xenotransplantation, giving humans organs from non-humans, introduces their viruses. And big changes in society with regard to drug abuse and sex. Uncontrolled urbanization, daycare centers, hot tubs, improperly sanitized hot tubs lead to infections. Air conditioning, water cooling towers with all that water spraying down and getting into the building. Legionnaire's disease, millions of used tires that have a little bit of water in them and breed mosquitoes and carry them around the world. And of course, don't forget, climate change is shifting where vectors are and their hosts. And natural disasters also. A hurricane can make a big difference in the kinds of mosquitoes that are around. So we are doing all these anthropogenic activities We to promote virus emergence. I love this. Humans continuously devise new ways to encounter viruses. Here's an example, the Amazon North region of Brazil. As you know, this is being deforested at a huge rate, and it is full of viruses. All these words are the names of viruses that have actually been isolated at the Evandro Chagas Institute in Brazil. And there are many more out there. And of course, the workers are at risk for getting infected. And, it's not something you're going to see every day, but it may take years for this to spread in the population. The point here is that there are lots of viruses out there. So here's a list of some viruses and the factors that led to their emergence. And we will talk about some of these today and next week. So we have the virus here, the family, and the factors leading to emergence. So, for example, dengue, we have the used tire trade. For Ebola virus, human contact with a natural host. Hantavirus, agriculture, human-rodent contact during the rice harvest. Hantavirus, proximity of fruit bats, favors transmission to horses and stable workers. HIV-1, hunting and butchering of infected primates. And, of course, influenza virus has a reservoir in aquatic birds, and bird and pig farming has expanded that, the spread of those viruses. MERS coronavirus, camel husbandry. Nipah virus, fruit bats near piggeries, in piggeries, actually. SARS-1, open-air meat markets. Sin nombre, increase in the deer mouse population. West Nile virus, mosquito from a bird reservoir, global travel helped spread that. And Zika virus, again, mosquitoes and global travel. Oh, down here we have SARS-CoV-2, a live animal market. That's going in the textbook, folks, not in lab origin. This is going in the textbook, live animal market, because that's where it came from. So what's the role of evolution in the emergence of viruses? What does evolution do? Remember quasi-species we talked about last time? Every virus stock is a collection of different genome sequences. So these are all mutations in each genome. And a pig that's infected with influenza virus has a quasi-species in it. And so what does that mean? Well, in that quasi-species, and it's much more than is shown here, there may be accidentally a virus that could infect another host should it get to another host. And so that's what happens. You have every animal in nature, no matter what it is, it's infected with something and it is a quasi-species. And if that animal contacts another, maybe there's one virus in that population that can infect just by random mutations in the viral genome. So that leads to adaptation to new hosts and environments through selection of these quasi-species. We talked about that a bit last time. So there are a number of ways that hosts and viruses interact. Let's talk about those for a minute, okay? Here in this picture at the top, we have a stable host-virus interaction. So this is a kind of interaction that maintains the virus in the ecosystem. So think about measles virus and polio virus and herpes viruses, okay, in humans, and there are lots in animals as well. Then we have an evolving host-virus relationship. And here's when virus is introduced to a naive population. It could be the same host or a different host. So the virus could go from a non-human animal to a human, and then it evolves for some time as it adapts to the new host. It could also go human to human, as we'll see when we talk about colonization of the new world. The dead end is a one-way passage to a different species, like West Nile virus is a dead end in every host because it cannot be transmitted to another. Technically, Ebola virus is a dead end because it never sustains human-to-human transmission. It can transmit a lot, but it's easily stopped, and it has not become a human virus yet. Same with influenza H5N1. And then finally, we have the resistant host where infection is blocked. The virus can get in that host. Nothing happens. No reproduction, no infectious virus produced. And these four different categories cross-feed each other. So, for example, viruses in a stable host-virus relationship can go into a new host and become an evolving infection. That's an emerging infection. And the same thing from a stable to a dead-end pathway. That is like West Nile virus is in a stable virus-host interaction in nature. And we'll see an example of that, and then it's transmitted to a dead-end host. All right. So in the stable host virus interaction, both participants survive and multiply. So one doesn't wipe out the other. So measles, herpes viruses, influenza virus, poliovirus, and so forth. And some of these are permanent. So we're sole hosts for these viruses: measles, herpes simplex, cytomegalovirus, and smallpox virus. And they are permanently human viruses, and they do not infect any other species. But of course, SARS-CoV-2 is not exclusively human. It infects many other animals. And often this infection, this stable, can include more than one species. So influenza A viruses, flaviviruses, togaviruses all infect humans, but they also infect other species as well. And in both, they can have a stable host virus relationship. The evolving host virus relationship is unstable and unpredictable. And sometimes the outcome is death, sometimes the outcome is a benign disease. So examples are the introduction of smallpox and measles to natives of the Americas by Old World colonists and slave traders. Those individuals had never seen that virus. They were immunologically naive, so it caused great damage in them, and that's an evolving infection. The introduction of West Nile virus into the Western Hemisphere, when the virus first came in, it spread across the country, changed quite a bit. It evolved as it got used to its new host. And of course, the introduction of myxoma virus to eliminate rabbits in Australia, which we talked about last time, if you remember. That was introduced into the rabbit population, and it evolved. Both the host and the virus evolved. So classic evolving host-virus relationship. And then we have the dead-end interaction, a frequent outcome of cross-species infection. There's no sustained transmission. Sustained is the key. Now, I know some of you are saying, oh, there's a transmission here and there with H5. It's not sustained. That's the definition. All right? It has to be sustained like SARS-CoV-2 is sustained transmission among people. And so no sustained transmission from the new infected host to others of the same species. Ebola virus is an example. In humans or chimps and gorillas, no sustained transmission. Yeah, there was a lot of transmission of Ebola virus in the 2015 West African outbreak, but there were other reasons for that. It never became a human virus. Once we intervened with infection control, proper infection control, we stopped it because it didn't become a human virus. Influenza H5N1 is another example. So these dead ends don't contribute very much to the spread of the natural infection, obviously. It's the stable and evolving that contribute to the spread. So let's take a look at some examples of stable and dead-end host virus relationships. So here is a virus that is spread among birds by mosquitoes. So we have a mosquito that is, taking a blood meal on a wild bird and then can bite another wild bird, and you have this cycle continuing in nature. The mosquito is fine. The virus reproduces in the mosquito. The birds are fine. Occasionally, the mosquitoes bite chickens, and the chickens get infected. Or occasionally, other mosquito species bite the bird, and then they feed on other hosts. So this is the stable interaction that exists in nature. Occasionally, these mosquitoes will bite a horse or a human, and they will introduce virus into them, and those animals may get sick, they may die, but they do not transmit it to another person. They don't have enough virus in them, in the blood, so that if the mosquito did take a blood meal from them, it wouldn't be enough to transmit to another host. So the mosquito biting the human, that human gets sick, and that's the end of it. Same with the horse. Now, it's not to say that these aren't serious outcomes, but there's no sustained transmission in humans or horses. So here's another one. This is a tick-borne virus, tick-borne encephalitis virus. Here the cycle is maintained between ticks and rodents. The ticks feed on the rodents. They get viruses that are, permanently established in the rodent, and then they transmit it to another rodent. And occasionally the ticks can bite a cow, in which case the virus may be present in milk and it will spread to humans. If you don't pasteurize your milk, folks. And sometimes a tick will bite a person, and these individuals will get dead-end infections. They will not transmit it to anyone else. Even if you took milk from a human, an infected human would not transmit. They're dead-end hosts. So that's the relationship between a stable and a dead-end relationship. So how do emerging infections begin? There are two steps. Right. We already said that in the animals in nature, they're quasi-species of viruses reproducing. Now they have to encounter something to spread it to. So bats living in a cave infect other bats. That's fine. But for a bat to infect a human, the bat would have to encounter a human, or an infected camel or an infected rodent would have to encounter a human. Now we know this happens, right? People keep camels as pets. They encounter them. Mice invade our homes and leave droppings that are full of virus. And bat-human is harder. We do know that people in the country, in parts of China, are infected with bat viruses. We know from serology studies. But often the bat infects another non-human animal, which is then brought to a market, and it infects humans. So then you have to, this virus has to encounter a human, and then it has to get established. It has to reproduce in that human, and then the human has to encounter other hosts to spread it. Because if that human is the only one that's infected, that's the end of the line. It's never going to go anywhere. It's never going to become an emerging infection. And so I think we have lots of chance encounters of viruses with new hosts all the time. We don't detect them. They're either dead end and not pathogenic, or the host is resistant. And even if a single human is infected, it may not be transmitted for many reasons, right? Because the human may not contact anyone during the time of shedding, might not be shedding enough virus, and many reasons like that. And we know that sometimes this works. SIV came from chimps to humans and established a new virus in humans, which we now know as HIV. And I think we have talked about the two pandemics before. Let me look at my list of lectures and see. The infectious cycle, antibodies. Yeah, we haven't talked about Tale of Two Pandemics. Oh, here it is. Tale of Two Pandemics was lecture number 8. So we already talked about HIV and SARS-CoV-2, COVID. But examples, there are cases where these rare encounters do spread and extend through the population, and they require specific circumstances, including, a highly populated city like Wuhan at 12 million, which is a transportation hub. It's a perfect scenario for a virus to begin and infect many people. Now, when these viruses that we've encountered from another animal enter us, they need to have susceptible and permissive cells. Remember, a susceptible cell has a receptor for the virus. A permissive cell is internally able to support virus reproduction. So that virus that gets in you has to find those cells and reproduce in them. and then spread. And so population density and health of the individual are important factors. Perhaps a not healthy person would produce more virus, and it's more likely to spread. And of course, population density is essential because that allows extensive initial spread of the virus, as we saw in Wuhan, China. You have to have serial infections, right? You have to go from an animal to a human and then to multiple humans to establish a chain of transmission. Otherwise, the viruses will not endure. It will just be a dead end. One person infected or a few. Let's take an example. A farmer who's living alone in a rural area gets infected with a bat virus. That's it. That's the end of the story. But if the farmer has a family, he can infect his family. The family can go to other places, schools, markets, and so forth, and spread the virus to others. So you see, we need this connectivity for it to get going. So let's now, for the rest of today and next week, let's talk about some of the origins of human viruses, where we know they came from. All of them, remember, from other animals. So here's smallpox virus. So by studying smallpox virus and related viruses, we build phylogenetic trees to see the relatedness. We can suggest that this virus emerged 3,000 to 4,000 years ago in East Africa, transmitted from camels. So here's East Africa right here, this circle where we think it originated. And camels were actually introduced to Africa 3,000 to 4,000 years ago. So camels originated in North America, and then they traveled south, and they traveled across into Asia, and eventually made it into the Arabian Peninsula. And we think the camels actually got infected with a smallpox-like virus from gerbils. So the gerbil range is shown here in this hatched oval. So the camels come here. It's a new environment for them. They encounter gerbils, which give them a virus, and that the virus percolates through camels, and then people get it from camels because they're using camels and they have close relationships with them. This is all what we think based on nucleotide sequences, of course. It's too long ago to really know for sure. Measles virus is closely related to a virus that infects cows called rinderpest virus. And we think measles emerged from an ancestral rinderpest virus when we began to domesticate camels in the 11th and 12th centuries, right? So cattle were out there in the wild, and people, would periodically catch them, and they found that they were good for milk and for meat. So they began to domesticate them. They grew them and had pastures full of cows, right? And the cows had their viruses. And so they gave us their virus, rinderpest, and that became measles virus. Now, this is interesting. Measles virus requires large populations to be maintained. And so when human populations began to congregate in cities, then we began to get transmission of measles virus among humans. So it established in the Middle East initially, measles virus, and then it spread around the world as infected colonizers and migrators went all over the world, and it reached the Americas in the 16th century and basically destroyed most of the Native American population. So that's measles virus. Now, this brings me to a little discussion of diseases of exploration and colonization. So here we're talking about a virus entering a completely immunologically naive population. So, for example, smallpox virus, it reaches Europe from the Far East in 710 AD. We just talked about where it originated. Gets to Europe, causes epidemics left and right, lots of people killed. And then the colonizers who went to the New World, some of them were infected. They brought the virus with them, and in the New World, nobody had ever seen smallpox virus before. And so in 2 years, after 1520, it killed 3.5 million Aztecs, spreading from Hispaniola, and that allowed conquest by Cortez with his small band of soldiers. They were immune to smallpox virus. They'd probably survived infection back in Europe, and maybe someone was infected or a few people were infected, and that wiped out the Aztec population. So it really changed the balance of human populations in the New World. Then changing human populations and environments. Of course, we've told this story before. It's the story of poliovirus, but it never hurts to repeat something, and this is probably the last one we'll talk about today. Poliovirus existed as a sporadic infection for many thousands of years. This Egyptian carving from a few thousand years ago shows a priest with what we think is polio. It has the drop foot typical of polio. But there were never any epidemics of poliomyelitis. And you can see in this epi curve that around the turn of the 20th century, suddenly we had outbreaks of many cases of poliomyelitis. So here's the year on the x-axis and the reported cases annually on the y-axis. So, if you didn't have enough in previous years to make a blip in this chart, but suddenly in the early 1900s, bigger and bigger outbreaks. And so why did the virus change from being a sporadic infection to an epidemic one? It's not population, because the population was relatively similar over these years, but it was rather the sanitary environment. So pre-1900, poliovirus circulated freely and infected most babies shortly after birth. We had no toilets, we had no sewers. We would use chamber pots, and I say we because it's the royal we. I never did it myself. You would use chamber pots, and then in the morning you would dump them outside. Often on the street, if you're in a city, the streets were full of feces, horse feces and human feces. Yeah, they got shoveled up eventually, but kids got infected because their mothers would go out and get contaminated and infect the kid. But the key was maternal antibodies were given to the baby during fetal development, and they protect the baby from poliomyelitis until about 6 months of age. And by then, when they get infected, they have a mild disease, non-paralytic, and they develop their own immunity. And what changed, though, after 1900, we developed the toilet and the sewer. We improved sanitation, so we delayed infection until children were beyond the point of having protective antibodies from their mother. And so now you have large numbers of kids accumulating who are seronegative. You put a virus in them. It's just like the colonists putting smallpox in the native population. You put poliovirus in a group of several thousand kids, boom, you have an epidemic. So that's how the epidemiology of poliomyelitis was changed by modern sanitation. Now, modern sanitation is good. So we developed vaccines against polio to prevent poliomyelitis, and they work very well if you use them. And I think we're going to see this ACIP eventually withdraw polio vaccines, as well as Hep B and HPV and basically all the others, and we're going to return to outbreaks of poliomyelitis, sadly. Okay, one more, yes. Changing climate and animal populations. Hantavirus pulmonary syndrome, a severe respiratory disease, first seen in the Four Corners area of New Mexico in 1993. So that's here where the 4 states come together. Cases of young individuals suddenly getting severe respiratory disease and dying. So the CDC went in and figured out it was caused by a virus endemic in the deer mouse. Paramyscus maniculatus, 30% of wild Paramyscus deer mice are positive for this virus. So the name of the virus is now Sin Nombre. It was originally called Muerto Canyon virus because that's where it was isolated. But the residents of Muerto Canyon didn't want a virus named after them. So the CDC, in a rare show of humor, called it Sin Nombre, no name. Anyway, the virus is a bunyavirus, an RNA virus, negative-stranded, enveloped RNA virus with 3 genome segments. So why did this suddenly emerge in the U.S. in 1993? In that year, there was abundant rainfall, and that led to a big crop of piñon nuts, which humans love, and so do the deer mice. And the deer mice ate the piñon nuts, and they expanded their population, and the contact with humans increased. They invaded their homes. The virus is excreted in mouse feces, and contaminated blankets or dust from floors were what infected humans. So the mouse defecates, the virus is present there, and then you try and clean up the poop, and you aerosolize it and inhale it, and that gets you your infection. So this is a zoonotic infection. Humans are not the natural host. Human disease is actually rare. And it does not transmit from human to human. So this is a dead-end infection. And when we studied this virus, and after its first isolation, we found that there were cases going back to 1959 in the U.S. We just didn't know what was causing it. But now in 92, 93, we're able to figure that out. And so if you look at Hantavirus pulmonary syndrome by state, this is through 2022, there are 831 cases. You can see most of the western states have most of the cases, although we have some here, sporadic, a couple of cases in New York and New Jersey. The deer mouse is not the only host. This is the host range of the deer mouse here in green. But you can see that the white-footed mouse, the rice rat, and the cotton rat are also hosts for hantaviruses. So if they get in your house, they can spread it too. So you should just be careful. If you see mouse poop, spray it with some Clorox first, okay, to inactivate the virus. Clorox is great at inactivating viruses. And then, sweep it up, and you'll reduce the chance of aerosolization. But as you know, Gene Hackman and his wife had an encounter with Hantavirus pulmonary syndrome. So they were living together in Santa Fe, and he was 95. He had Alzheimer's, and he was dependent on his wife, who was 65, for care. She died of hantavirus infection, and then he had no one to take care of him. So she just died in the house and was lying there. And he died eventually 7 days later because he couldn't feed himself. So they were both found dead in their home. So she died of Hantavirus pulmonary syndrome. Presumably mice invaded their home, infected her, and then because she wasn't around, he died as well. Very sad story. Okay, so that's part one. Next week, we'll go through emergence of other viruses that you know and love, like Hendra and Nipah and SARS-1 and SARS-2 and Ebola and more, okay? All right, let's shut that off. Back to the questions. Let's see, I put a star. Go all the way up. Good, you guys are asking questions. I learned a while ago to put a star next to the last question I answered. That was Pete. Okay. Okay, Charlotte says this is the AMA. Charlotte had said there was an AMA update, and they talked about the firing of the vaccine experts. They thought it was a bad idea. Yeah, it is a bad idea. As Paul Offit said, they have lost the trust of the American medical establishment. And that's why people are going to start setting up their own advisory boards. John says, emerging viruses, what's old is novel again. In some cases, yeah. In some cases, that's true. Yeah, Fiona Havers, right. That's the article, and she's the CDC person who resigned. The article just points out all the great things she did. Kevin, how many virions does the body make over a single infection? Depends on the virus, right? But let's say a respiratory infection like influenza virus. A single infected cell can make thousands of virus particles. So if you have millions of infected cells, millions and millions of virus particles a day over the course of days, tens of millions of virus particles, most of which never make it in the world. They never find a new host, but perhaps a few will if that person is in contact with other people, right? Does the follow-on reinfection generate less total virions? Excuse me a moment. Well, if you've generated good immunity to the virus of the first infection, yes, the load should be reduced. It is not eliminated, right? No immune response is sterilizing, but it is reduced. And so that could result in less severe disease, mild disease, and less transmission. Yeah. So is the first infection most often the worst? Yes, unless the immune response is part of the pathology, right? So the first infection, you're naive, and it takes you 2 weeks to make an immune response. Now, there is an innate immune response very quickly within hours, and that saves most of us. I think that's responsible for curtailing most infections. But if it gets past that, you need 2 weeks to make antibodies and T cells. And so that first one would be the worst, yeah. And so if you're compromised, if you're elderly and have comorbidities, and you get infected with a virus for which you are immunologically naive, it's toast for you. That's why so many elderly people died during the COVID pandemic. Nevitz says we are always doing dangerous things. Drill, baby drill, climate change later, plastics for everyone, and 60 years later, microplastics everywhere. And what about the forever chemicals, right? Like slippery frying pans, and now we have forever chemicals that are around forever. Yeah, how about that? I guess measles is a very ancient virus, so it's adapted and specific to humans. So yes, measles virus over the years it became adapted to humans and lost its ability to infect cows and other. And there are different kinds of measles-like paramyxoviruses that can infect other species like dogs. But yeah, it's a human virus now. Just like polio has become a human virus. SIP3 says, it hurts to see you not be loved for the work, the endless passion for science you have shown and taught so many of us. It has been a true honor to have these years with you, bud. What, are you leaving? Because I'm not. Not going anywhere. Visto, good question. To sustain in humans, would H5N1 influenza need to replace the H5 with an H1 or H2 or something? This is a really good question. So what would it take for H5N1 to be, transmissible sustainably in humans? So yeah, if you put an H1 or an H2, that would do it or could do it in theory. But, the H5 hemagglutinin has properties that make the virus particularly nasty. So it has a furin cleavage site in the HA, and that cleavage is needed for infectivity of the virus. And furins are present in pretty much all tissues in our bodies. And so that means H5 viruses can reproduce everywhere, whereas all the other ones, they're limited to your respiratory tract because it's a different protease that cleaves the HA. So if you replace the H5, you're going to lose that. It's going to become less pathogenic. The other possibility is that H5 changes to be sustainably transmitted among humans. Okay. Now there were experiments done in ferrets years ago that asked what kind of changes in the virus would allow aerosol transmission. So the H5 virus doesn't transmit well among ferrets, and several labs were able to produce a virus that could transmit among ferrets. And by the way, that stirred up a whole storm of controversy. And they identified changes that could allow that, but we don't know what's needed for people. I mean, you definitely need to bind human receptors. But what else do you need? And, it's been circulating for ages, and that hasn't happened, which could mean that maybe H5 can't change. It would break if it became transmissible as part of a virus. So, yeah, those are the two possibilities. Yeah, we just don't know. So Thought Seer says it began with the first farm animal, Greek pigs. Ironically, the first Western hospital also began in Greece and was at Epidaurus. It was founded on the highest mountain. I'm going to Greece in September. Thessaloniki. Bad Cat had to use AI, Chat GPT, to understand the discussion on Tweevo this afternoon. This little, very tiny archaeal microbe. And Logotic is ordering Fever. Fever's a great book. It's cheap. Go for it. Kim, yeah, Fever is a fun read. Polka Dot Pup. Can people get viruses from plants? Generally not. There is one example of a virus that is thought to have come from plants via insects, but a lot of people don't believe it. So I haven't seen any other examples of that. So I think they're just too different. Hello, Jessica from Toledo. Welcome to Office Hours. Visto says viruses just love mammals that can fly. Bats and now humans. That's right. Of course, viruses don't love anything, right? Mm-hmm. Yes, folks, hit the like button. There are 145 of you and just 107 likes. So just hit it, and it may attract some other people here. Who knows? Les says we have to get the animals to wipe their feet and wash their hands in the wild. We can't even get humans to do that. Well, maybe animals would be better behaved. Who knows? Nevitt says, I'm afraid much of this will be barred from being taught at schools. I love the poorly educated. Well, the kids can watch YouTube, right? Can watch these programs and my lectures. Maybe that's a way to get around it. John gets the image of teaching little frogs to wash up. How cute. How cute. Here's an interesting idea. Viruses in the lab are pretty much like wingless pigeons. They don't get far and don't represent valid examples for transmission research either. Yeah, it's all artificial. And this is true. They don't contribute anything to the natural history of viruses. Do the bat viruses that make us ill make them ill? Do they all carry the same virus, or is the spillover just one unique virus that got very lucky after arising in one bat? Well, this is very complicated because there are many different species of bats, and often they have populations of viruses that are unique to each other, but bats often co-roost. Different bat species often roost in the same caves, so they can spread viruses that way as well. Okay, so do they make them ill? The prevailing notion is that they mostly don't get ill, but I don't like the blue. It's uneven. Let's fix that. Thank you. But it's hard to know because who has sampled enough bats to know if they get sick or not, right? I suspect that sometimes they get sick and sometimes they don't. People have done studies of bat virus populations over time, and you can see there are pulses of virus production that correspond with stressful things like migration or pregnancy, right? So they're not continuously infected at high levels anyway. So these pulses in the population. But, I think they get less sick than other animals because they have an interesting immune system, which has been developed to deal with oxidative stress of flying, right? But I think it's hard to say that they're all okay. Now, spillover. It could very well be that a virus arose in one bat, but that bat has to infect another animal, which is then taken into market, right? So in the Huanan seafood market, there were no bats for sale. Other animals were found to be contaminated with SARS-CoV-2. So it could be one, it could be multiple bats. I mean, I have a hard time thinking it's just one bat and one virus in a single bat. It could have been a population of bats where the quasi-species contained one or more viruses that were able to infect humans and just needed to go through another animal. Hard to know. We don't have enough data on this, but they're all good questions, very good questions. Massive air travel with COVID being infective asymptomatically. SARS-CoV-2 had at least a month to travel the world before it was detected, if a pandemic was inevitable. Well, this is the reason why it was such a great spreader, because, 40% of people are asymptomatically infected. So they're shedding virus while they're walking around. And that's the perfect property to spread. And yeah, it did take a while. I remember looking at the COVID world map with the dots on it, right? We did this in my virology class. Before every class, I would put that up on the screen. And for ages, it seemed to be just in China, then surrounding countries. And then it took a long time for it to get to Europe and then finally to the US. And I think all of those countries had people infected already, but we didn't know it because so many were asymptomatic. Yeah. It had to get to a certain level before we saw the disease. But yeah, it was air travel that spread it for sure, right? Kevin asks, is human to animal spillback infection to a different animal which continues in that animal species considered a zoonotic infection? Well, we call it reverse zoonosis, right? Oh, there's another word for it with anthropogenic in it. Let's see if I can find it. Most people, anthroponosis. There you go. Anthroponosis, or reverse zoonosis. That's the name. It's not a zoonosis. It's just to distinguish it. We are all animals, but the zoonotic term from animal to a human, that's weird because we're all animals. But the zoo, it implies what you might find in a zoo could infect you. A zoonosis, animals. Quasi-species is another great name for my next band. Yes. Well, I have one too. I think the other day I thought of one. C1q and the collectins. These are members of the complement pathway. We have a section in our book called C1q and the collectins, and I think that has a nice ring for a band. But quasi-species is another one too, yeah. There's a Cambodian rock band called Dengue Fever. They're actually pretty good. Check it out. Okay, Kang, is fitness for viruses a catch-all term for improvement of attachment, entry, replication, assembly, release, or all the above plus other factors? So the way I look at fitness is increased fitness means improved reproduction, more virus particles. And the reason can be any of these things that you mentioned: attachment, entry, replication, assembly, release, and other things as well. But all of those lead to improved replication. So increased fitness can be looked at simply as improved replication. So here's a good way and a good example. We have poliovirus Sabin vaccine strains, the attenuated strains. They have reduced fitness. They were selected by Albert Sabin to have reduced fitness. But there's great pressure when those viruses are in your gut to revert to higher fitness. So the Sabin strains generally don't cause paralysis because they don't make enough virus to get into the CNS. But once the fitness is restored, those viruses then make higher levels of virus. They have higher viremia, and they can get into the CNS in an unimmunized person. Don't forget that many were infected in Las Vegas at the CES exhibition, which hosted 11,000 Chinese among its 170,000 visitors. Yeah, these kinds of events are super spreader events, is that they introduce infected people into a naive population. Remember, we were all immunologically naive, no impediment at all to virus spread. In Australia, there's a kids show where the rabbit's character's name is Mixo. That's great. Melinda, of course, that's what office hours is for, to ask virus questions. If we know, was measles present in the Americas before the 1492? Let's look it up. So the earliest epidemics are recorded in the early 1700s. So it wasn't present after that colonization. Melinda wants to know, what's the impact of acute COVID for someone who already has long COVID? Well, it could have no effect, or it could make it worse. So multiple infections sometimes make it worse. It depends on factors that we don't understand. Never kiss a camel on the mouth. Absolutely. In order to mount an immune response to a vaccine, we use vitamin D. A lot of people are D deficient or insufficient and don't know it. Why don't we recommend taking vitamin D before vaccination? Yeah, that's a good question. I had a blood test for vitamin D and I was deficient, so I started taking vitamin D because I was inside all winter and spring, right, getting no sun. I don't know why it's not recommended. Maybe it's not thought that it's worthwhile. I just don't know, Melinda. It's a good question. Polka Dot Pup asks, if a rodent is in a shed and leaves droppings and has a virus, is the virus active forever, or does it eventually become inactive? It will eventually become inactive, right? As the poop dries out. Most viruses don't survive desiccation, and so maybe within days it's no longer infectious. But, you see some fresh mouse poop, your inclination is to clean it up right away, right? And then it's still fresh. Is there any progress program to eliminate herpes simplex from its human host? Yes, people are exploring different approaches, trying to cut out somehow the viral genome from the neurons, the peripheral neurons where it's located, using something like CRISPR-Cas and a gene drive where you put a virus in them and it drives through and cuts out all that. But we're a ways off from being able to do that. Don't kiss gerbils either. Poliovirology, yep, that's part of it. And we will look, we talked a little bit about that last time in evolution. Questella is camel-free. And John says when he quit smoking, he also became camel-free. John is on a roll tonight. How often do animals die from COVID? Well, deer don't seem to have any problem at all. But, and rodents don't seem to have it. Domestic animals, more of a problem. I don't know why. Maybe, I don't know, not sure why that is. Has this changed over time? No, mink farms got infected. They got sick. I don't believe it's changed over farm. Costella's kid just said he thought measles came from weasels. Weasels. Mustelids. Tell your kid that it's a mustelid, like a ferret is a weasel. But not all weasels are ferrets. Maybe if we hadn't used camels for meat, they wouldn't have given us their viruses. Well, but people don't just use them for meat. They're pets, and they are used for racing, so there are other uses as well, right? Bus Drive and thoughts for TWEVO from bioRxiv, a cellular entity retaining only its replicative core. We just did that paper on TWEVO today. Good, Bus Driving One, you're on it. Thank you, Mark. I'm glad you like it. I just love viruses, as you know, so it's infectious. Mark says, I thought hemolytic smallpox caused the Adenine plague in the Roman Empire. I don't know. Let's look it up. See, 165 to 180 CE. Possibly smallpox or measles, according to the interwebs. Polka Dot Puck also likes tonight's lecture. Thank you very much. Hantavirus killed a grad student I knew in the 90s. She got it in Tahoe. That's too bad. Yes, it can be very bad. With sanitation leading to the polio epidemic, are you suggesting we all eat feces again? No, that's disgusting. Just get vaccinated. I know you're joking. It's amazing how much John encountered the term oral fecal, learning about infectious disease. Rita loves the subject. I am interested in how pathogens interact with the natural world. Kip says, in the expanding niches slide, shouldn't the black dots have two-sided arrows between them? Let's take a look, Kip. All right, let's reopen it. Expanding. Expanding niches slide. The black dots. Oh, I see. Rows of evolution. I don't know which one you mean, John. Kip. Sorry. Tell me what number it is, and I'll look at it. John, I saw a cute moment of a doe coming up to a man and his dog on the sidewalk, and although he was pulling out his phone to record, all I could think about was zoonosis. That's right. But you do know that it's rare. These zoonotic events are extremely rare. But nevertheless, caution is advised, yes. Tom, Hantavirus, pack rats can be a host. Be sure to use fresh bleach. I didn't realize that commercial bleach has a relatively short shelf life. We used it. We used it all the time in the lab to inactivate poliovirus. Visto asks, if human civilization lasts for 1,000,000 years, would we get spillover of all the viruses eventually? No, it's probably not enough time. There's a lot of animals out there and a lot of viruses. What mechanism, Melinda asks, does the body use to clear virus parts after the immune system kills the virus? Oh, the viral particles are broken up in cells. They can be broken up in like macrophages, and then the parts are recycled. And even virus particles that are inactivated and floating around, they're taken up by these phagocytic cells and then they're destroyed and recycled. Yeah. Javier wants to know, what techniques do we use to identify genes in the human genome with viral origin? Do we just look at any overlaps between the human genome and virus genomes and make an inference? So it's not easy, as you might know. So we have human genome sequences, and we have lots and lots of viral sequences, known viral sequences. And so, the simplest thing is just to do a search, use a computer to take all the viral sequences and search against the human, but that's going to be full of noise. So you have to do more sophisticated approaches, but they're all computational. And sometimes the homology is very small, so you have to tweak your approaches to make sure. And then in the end, you can see the sequence, whether it's viral. And many people have done that, and you can see genes, particularly retroviruses integrated in the human genome. You can see that those are of viral origin, but there are also non-retroviral sequences integrated as well. Yeah, the first papers, they just did what we call a blast search, right? You use a computer program to compare human and viral sequences. Clorox is premium and lasts longer. Yeah, we used to buy Clorox. If you buy it from a scientific supply company, it's $45 a gallon. So I used to go to the supermarket and buy it for $10 a gallon and bring it in. Thank you, John, for your support of science communication. Extra episode this week with David Tuller, MECFS and long COVID. Corey wants to know, why do the RNA polymerases switch to creating the whole antigenome instead of just making mRNA? Because the mRNAs are short and they cannot program a new virus. The viral genome is longer, and you need that extra sequence to put in a virus so that when that virus infects the cell, it will make new viruses. If you put mRNAs in the viruses, the infection wouldn't work because the mRNAs are too short. That's the quick answer for that. Yeah, in minus RNA viruses, that's what goes on. Angry Penguin wants to know, apart from insects, are there any non-mammals like reptiles that have transmitted viruses to humans? No reptiles, no amphibians, no plants, no fishes. No, not to humans. Mostly it's mammals, and sometimes the vectors can be insects. Yeah. Yeah, birds. Birds can do it too, right? I always forget the birds. Hello, Danny. Welcome from Nova Scotia. Yeah, wearing a mask would help too. Yep. Miss Martian's friend of mine, child, died of Hantavirus when I was there in New Mexico. It's still present all over the state now. Isn't there a virus people get from potting soil? I never heard of that. You could get Legionella from soil, which is not a virus, of course. Right? Fever 1793. No, it's just Fever by John Fuller. Okay. Kevin, this is a tough question. Why did COVID settle into a bimodal 2 waves per year infection pattern? Why doesn't flu do that? I don't know why COVID is doing twice a year. I think it's weird. I think it should be a winter respiratory virus. By now, most people got some kind of an immunity. Maybe that's going to take care of it. I don't know. So I don't know the answer to that. And I don't know why flu does one. Will measles become bimodal? No, before vaccinations, measles was not bimodal. It's just SARS-CoV-2 that has this weird thing. Why can't rabies transmit via the bloodstream? Well, I don't know how you would do that. I mean, in humans, rabies is relatively rare. So intravenous drug use, but a person who has rabies is not going to be doing that. And so I don't know how else it would get one person to another via the bloodstream. China is having a huge increase of COVID. The government says it's mild. Well, yeah, it's just one of many cold viruses now in most people, right? I don't know that it's worth tracking, but you're going to have seasonal increases, right? Yes. Danny says Neanderthals must have encountered bad viruses. Early hominids lived in caves, maybe ate bats. Yeah, maybe. It's possible. We just don't know any of this stuff, right? Yeah, mild COVID still causes a certain percentage of long COVID. I don't know about the spike protein involvement in it. I'm not sure I buy any of that. If there was another spillover, could an mRNA vaccine be developed easily? Not in this climate. RFK wouldn't let an mRNA vaccine be made. He wants to get rid of the mRNA vaccines that we have for COVID. He thinks they're bad, which is wrong. There's no evidence that they're bad. They're very safe. Safest vaccines ever tested. So I think it's going to be hard to make new mRNA vaccines. In theory, yes, you could develop it quickly and easily, but I have doubts. Jessica works with Flavies. Call her band NS5 and the Vectors. Yes, that's very good. I like it. Evitt says Alzheimer's mRNA vaccine phase 1, no negative side effects, 3-fold higher levels of antibody against amyloid B. TWIV? I don't think so. That's This Week in Neuroscience, right? Because this is not a viral thing. Okay, let's see here if there's anything else we should... here's a good one. If there's so many mutations in a few kb genome, why do not become functionless? Because there's an error threshold. For RNA viruses, it's quite high. You can exist below it, but as soon as you exceed it, then you're gone as a virus. We don't see those viruses, right? But it happens a lot. They're gone. They extinct because there are too many mutations. So they have to stay below. The viruses that we see are below that threshold. Yep. Yes, Heather, sorry, we're almost wrapping it up, but you can watch it later. 1531 measles in Mexico. There you go. Camelid spit in your face. Hello, Margot. 112 in Phoenix Metro. Wow. 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 Hello, Martha. Returned from the Philippines. I've had 4 zoonotic diseases. Oh, she's going to list them, Rita, is pork ascarid, salmonella, cryptosporidium, Q fever. Wow. Good for you. You're a tough one there. Why would you want to keep toxic virus parts in the body? Aha, because the immune system needs them. They're stored in the lymphoid tissues to make better antibodies by affinity maturation, but that's a longer story we don't have time to cover. All right, it's 10 o'clock. Is there anything else here I should thank? Okay. Yeah, this is the last one. An mRNA vaccine could absolutely be developed quickly, but it won't be in the U.S. Yep. All right, folks. Thanks for coming tonight. Let's thank our moderators. We had Steph, Les, Vanity Nutrition, and Tom. Thank you so much for keeping it civilized. Thanks to all of you. You still haven't hit the likes to equal the number of people here. If you haven't liked, hit like as well, please. We appreciate it. We will be back next week with part 2 of emerging infections. So that is June 25th. All right, folks, June 25th, we will be back with office hours 8 p.m. Eastern Time. Well, Joel, I have to thank Joel here. Joel just gave us some money for science communication. Or maybe he did before and I just missed it. Thank you, Joel. Appreciate your support of science communication and everybody else who contributes as well. Really appreciate it. Been a blast, folks. Thanks for your participation and your questions. See you next week. Meanwhile, stay safe. Goodbye.